Today I, Dr. Vijay Prabha from Department of Microbiology, Panjab University, Chandigarh, India, going to present my work on studies on microbial infections associated with azoospermia in male mouse model and multifaceted insights into the potential mechanisms of spermatogenic failure. Within the enigmatic realm of reproductive health, azoospermia stands as a silent challenger, casting shadows on the aspirations of countless individuals yearning for parenthood. Azoospermia is characterized by the total absence of sperm cells in samples of seminal fluid. This condition impacts roughly 1% of the male demographic and approximately 10 to 15% of males encountering issues with fertility. Azoospermia is divided into two types, obstructive azoospermia and non-obstructive azoospermia. Obstructive azoospermia or OA accounts for 40% cases, whereas non-obstructive azoospermia NOA is the more prevalent form, affecting approximately 60% of azoospermic men. Several studies have unveiled the underlying mechanisms responsible for the pathophysiology of NOA. However, despite these investigations, the etiology remains elusive for the majority of cases. To achieve successful treatment of azoospermia, it is imperative to possess a comprehensive understanding of the diverse factors that contribute to this condition. Current findings underscore the significant role of microbial infections in the development of OA as well as NOA. Although OA has traditionally received more attention, there are major unexplored avenues in the connection between microbial infections and NOA. Urogenital infections caused by bacteria has been implicated as one of the major contributors to the development of azoospermia. Previously in our laboratory, deteriorative effect of sperm agglutinating, sperm immobilizing, SA or SI microorganisms, viz. Escherichia coli, Serratia marcescens, Klebsiella pneumoniae, all sperm agglutinating, and Staphylococcus aureus, sperm immobilizing, on reproductive potential of male mouse model, leading to infertility has been observed. However, the precise mechanisms by which these sperm-impairing bacteria induce azoospermia remain poorly understood. In our study, we aim to shed light on the potential mechanisms through which these bacteria exert their deleterious effects on spermatogenesis. Hormonal dysregulation, especially on testosterone concentrations, can perturb the finely tuned processes of spermatogenesis, culminating in diminished sperm production and overall reproductive competence. Reactive oxygen species ROS play a key role in the development of spermatogenic failure and can damage the delicate processes involved in sperm production, leading to a series of problems that ultimately result in azoospermia. Also, as ROS levels surge, they catalyze oxidative damage to produce spermatozoa, thereby compromising their functional integrity and viability. Extant literature elucidates that elevated levels of ROS may jeopardize the structural integrity of the blood-testis barrier, thereby exacerbating spermatogenic dysfunction through heightened permeability and impaired germ cell protection. Based on the above, by delving into the intricate details and investigating factors such as alterations in the reproductive hormone levels and the generation of various reactive oxygen species within testes and other reproductive organs, we aim to uncover the specific mechanisms employed by sperm agglutinating and sperm immobilizing bacteria in disrupting the normal process of sperm production, leading to azoospermia. Microorganisms investigated. So we have three microorganisms under the category of sperm agglutinating: Escherichia coli, Serratia marcescens, Klebsiella pneumoniae, and under the category of sperm immobilizing, we have Staphylococcus aureus. Escherichia coli and Staphylococcus aureus, they are the clinical isolates, whereas Serratia marcescens and Klebsiella pneumoniae, they are the standard strains obtained from Institute of Microbial Technology, Sector 39, Chandigarh, India. The main objective of our studies was colonization of various sperm impairing bacterial strains in the genital tract of male mice by intratesticular administration and to understand the mechanisms of azoospermia. For these studies, we used the male biopsy mice and they were sexually mature, aged 5 to 6 weeks with an average weight of 25 gram. All experimental procedures were carefully reviewed and approved by the Institutional Animal Ethics Committee of Panjab University, Chandigarh via the registration number as given in the slide. For the present studies, all the animals, they were divided into 8 groups with 5 mice in each group. We had 6 groups under the sperm agglutinating category and 2 groups under the sperm immobilizing category. And under the sperm agglutinating category, in test T1 we administered E. coli, in test T2 we administered Escherichia marcescens, and in test T3 Klebsiella pneumoniae was administered. Whereas in test T4 Staphylococcus aureus was administered, whereas C1, C2, C3 and C4 they served as control groups. This is how the organisms were administered intratesticularly. All the animals were inoculated under the anesthesia of ketamine and xylazine, which was administered intraperitoneally. Under sterile conditions, the mice were placed in supine position and the scrotal skin was gently washed with saline solution and disinfected with 70% ethanol. The abdominal area was gently pressed so as to protrude out the testes. The male biopsy mice were inoculated with either a single dose of 20 microliter of PBS in the control group or 10 raised to power 8 CFU of each microorganism in 20 microliter of PBS in the test group. The point here to be noted is the organism was administered as a single dose only in the right testes. The next experiment was to determine the bacterial load in the reproductive organs of the mice administered with various microorganisms. Mice were sacrificed on 7th day by following the strict protocol of IAC-UC. Reproductive organs with testis, caudal epididymis and vas deferens of both the sides were carefully removed, that is right side as well as left side. Homogenates were prepared in 1 ml of PBS through hand homogenizer. Homogenates were centrifuged at 12,000 G for 10 minutes at 4 degrees centigrade and the supernatant was collected. Bacterial load was calculated in terms of log CFU per gram of tissue. As far as bacterial load determination is concerned, we can see from the table that in control group we could not get any bacterial count, maybe in the testes, maybe in the epididymis, maybe in the vas deferens, where the count was 0 both on the right side as well as on the left side. Whereas in the test group where the organism was administered, maybe E. coli, maybe Escherichia marcescens, maybe Klebsiella pneumoniae, we could get significantly high count on the right hand side as compared to the left hand side. In case of E. coli, in the right testes we could get 9.9 CFU. In case of Escherichia marcescens, count was 10 CFU. In case of Klebsiella pneumoniae, it was 9.0 CFU. Whereas on the left hand side we could get 5.8 CFU in case of E. coli, 5.57 in case of Escherichia marcescens and 5.5 in case of Klebsiella pneumoniae. Whereas in case of epididymis, again we can see that the count was higher on the right hand side as compared to the left hand side. In case of E. coli, it was 9.1 CFU in the right epididymis. In case of E. coli, 9.1 in case of Escherichia marcescens and 8.8 in case of Klebsiella pneumoniae. Whereas on the left hand side we could get 6.6 CFU in case of E. coli, 6.7 in case of Escherichia marcescens and 6.5 in case of Klebsiella pneumoniae. Whereas in case of vas deferens, again we could see the same pattern, count was higher on the right hand side as compared to the left hand side. In case of E. coli, the count was 8.6 CFU on the right hand side, 8.57 in case of Escherichia marcescens and 8.5 in case of Klebsiella pneumoniae. Whereas on the left hand side we could get 7.2 CFU in case of E. coli, 7.2 in case of Escherichia marcescens and 7.2 in case of Klebsiella pneumoniae. So from all these results it can be concluded that even if the organism is administered in the right testis only, but we can get the count even in the epididymis in the vas deferens both on the right hand side as well as on the left hand side, and the count is in the same pattern as the organism travels from right testis to right epididymis to right vas deferens to left vas deferens to left epididymis and to left testis. In case of sperm immobilizing organism also, we could get the same kind of results where the count was much higher on the right hand side as compared to the left hand side, and the count was maximum in the testes as compared to epididymis or vas deferens. In the testes, the count was 10 CFU on the right hand side, whereas on the left hand side the count was 5.6 CFU. In case of epididymis on the right hand side, the count was 9.2 CFU, whereas on the left hand side the count was 6.7 CFU. In case of vas deferens also, the count was higher on the right hand side, that is 8.64 CFU, whereas on the left hand side the count was 7.2 CFU. So from these results also it can be said that the organism got established in the higher number on the right hand side as compared to the left hand side, and that also maximum in the testis as compared to vas deferens. The next experiment was to confirm that the organism that colonized in the reproductive tract was the same as the organism that was administered. So the homogenates, they were streaked on different media. In case of Escherichia coli, homogenate was streaked on EMB agar, and we could get beautiful green sheen indicating that the organism colonized was the same that was administered. Similarly, in case of Escherichia marcescens, when the homogenate was streaked on nutrient agar, we could get red colonies, again indicating that the organism is same as it was administered. Similarly, in case of Klebsiella pneumoniae also, when the homogenate was streaked on the McConkey agar, we could get beautiful pink mycoid colonies, again indicating that the same organism has been established over there, the one that was administered. Similarly, in case of sperm immobilizing organism, when it was streaked on the mannitol salt agar, we could get beautiful yellow colonies indicating the organism administered is the same as that got established. And from these results we can also see from the control group that we could not get any organism when homogenate was streaked from the control group on nutrient agar, indicating that there was no bacteria that got established. The next experiment to find out the seminal parameters. Following the incision of the abdomen, the vasa differentia were separated from the testicles and epididymites using a magnifying glass. The clean vasa differentia, both from the right side and left side, were then placed in glass plate containing 500 microliter of pre-warmed normal saline at 37 degrees centigrade. To extract the spermatozoa, the vas differentia were gently squeezed. Spermiogram analysis was done with regard to sperm count, motility, and viability. Under the seminal parameters, we did the sperm count. We find out the percentage motility and percentage viability. For sperm count, a fixed volume of 10 microliter of sperm sample was applied to a glass slide and examined under a light microscope. Average number of spermatozoa in these fields was multiplied by a factor of 10⁶ according to WHO guidelines. For motility, 10 microliter of sperm sample, that was 40 into 10⁶ spermatozoa per ml on microscopic slide, was kept and examined under a light microscope. And percentage motile sperms was calculated, number of motile sperms per field divided by number of total sperms per field multiplied by 100. And for viability, 10 microliter of sperm sample was combined with 10 microliter of eosin solution droplet on a microscopic slide. Presence of pink stained cells, which is considered to be dead spermatozoa, and unstained cells indicating live spermatozoa, they were counted, and percentage viable sperms was calculated by number of live sperms per field and total number of sperms per field multiplied by 100. As far as the seminal parameter analysis is concerned, we could find from the results that in case of sperm agglutinating microorganisms, maybe E. coli, maybe Escherichia marcescens, maybe Klebsiella pneumoniae, all the three microorganisms could lead to complete azoospermia on the right hand side, as there was zero sperm count as compared to the control group where we could find 48 million sperms present. As far as the left hand side is concerned, there was a count of spermatozoa, but it was significantly less as compared to the control group. In case of E. coli, the count was 2.2 million. In case of Escherichia marcescens, the count was 4.2 million. And in case of Klebsiella pneumoniae, the count was 9 million. As far as the motility is concerned, it was found that in the control group, 86% sperms they were motile. But in case of E. coli, as there was complete azoospermia, therefore the motility could not be determined on the right hand side. Similarly, in Serratia marcescens and Klebsiella pneumoniae also, as there was complete azoospermia, motility could not be detected. Whereas in case of the left hand side, all these sperms they were non-motile, although there was count, but they were all non-motile. In case of Serratia marcescens, 7.7% sperms they were motile, whereas in case of Klebsiella pneumoniae, again all the sperms they were non-motile. As far as the viability is concerned, in the control group, 87% sperms they were viable, but in case of E. coli, viability could not be detected as there was complete azoospermia on the right hand side. In the left hand side also, all the sperms they were non-viable. Similarly, in case of Serratia marcescens, on the right hand side, viability could not be detected because of complete azoospermia, whereas only 4.7% sperms they were viable. In case of Klebsiella pneumoniae, in case of right hand side, viability could not be determined because of azoospermia, and on the left hand side also, all these sperms they were non-viable. In case of sperm immobilizing microorganisms, Staphylococcus aureus, again the count in sperm was significantly lower as compared to the control group, but it could not lead to complete azoospermia. So on the right hand side the count was 5 million, whereas on the left hand side the count was 21 million as compared to 48 million in the control group. As far as the motility is concerned, the motility was significantly lower as compared to the control group. Here again the sperms they were completely not non-motile. Here 31% motile spermatozoa could be observed on the right hand side and 60% spermatozoa they were motile on the left hand side. As far as the viability is concerned, 87% they were viable in the control group, whereas the 35% they were viable on the right hand side and 64% viable on the left hand side. So from these results it can be observed that sperm immobilizing microorganisms they are less efficient in causing the azoospermia as compared to sperm agglutinating microorganisms. These photographs show how the spermatozoa they look when they are dead and how the spermatozoa look when they are live. So in the first photograph A, these are unstained spermatozoa, that is a live spermatozoa showing no pink color, whereas in the B photograph we can see pink colored spermatozoa which are the stained spermatozoa and they are the dead spermatozoa. Next experiment we performed was the estimation of testosterone levels in the serum. For the estimation of testosterone, we collected the serum sample. On the 7th day of the experiment, blood was collected from the tail vein and it was allowed to undergo clotting, and then the supernatant containing the clear serum was collected, and then solid phase competitive chemiluminescent enzyme immunoassay using Imulite 2000 total testosterone assay kit was outsourced to Malhotra Medical Laboratories, Private Limited, Sector 22, Chandigarh. The level of testosterone in serum has been depicted in the following table, and we can find from this table that the normal range of mouse testosterone, it is between 1.7 to 14.4 nanogram per ml. In the control group administered mice, the level was observed 2.5 to nanogram per ml, which was within the normal range. However, in the mice administered with sperm agglutinating microorganism, the level was found to be significantly reduced. For example, in case of E. coli, the level was 0.09 nanogram per ml, indicating about 96% reduction from the control group. Similarly, in case of Serratia marcescens, the level was significantly low. It was 0.078 nanogram per ml, where again we could find about 97% reduction in the testosterone level as compared to control. Similarly, in Klebsiella pneumoniae also, about 96% reduction could be seen in the testosterone level. In case of sperm immobilizing microorganism, the level was significantly low, that was about 0.22 nanogram per ml, about 91% reduction. From these results, it can also be observed that Staphylococcus aureus, although did reduce the testosterone level, but sperm agglutinating microorganisms, they were more efficient in reducing the testosterone level as compared to sperm immobilizing microorganism. Next experiment we performed was the estimation of reactive oxygen species generation in response to sperm impairing microorganisms colonizing the male genital tract. Reactive oxygen species were estimated by MD level, catalase activity and peroxidase activity, whereas antioxidant activity was measured by reduced glutathione and superoxide dismutase. MD level was estimated by TBA-TC method And for this, we took the tissue homogenates of testis, epididymis, and vas deferens. And the mixture was centrifuged at 3500 G for 10 minutes to take the supernatant. Absorbance of the supernatant was measured at 532 nanometer. And the MDA level was expressed in terms of nanomoles of MDA per gram of tissue using the molar extinction coefficient of 1.6 into 10 to the power 5 per mole. And the results showed that there was significant increase in the MDA level in the testes, in the epididymis, and in the vas deferens. However, the maximum increase could be seen in the testes as compared to epididymis and vas deferens. And the more significant increase was in the right side as compared to left side. In case of E. coli, there was about 3.64-fold increase. In the left testis, it was 2.56-fold. In case of right epididymis, it was 1.70-fold increase. And in the left epididymis, it was 1.35-fold increase. And in the right vas deferens, it was 1.46-fold increase. And in the left vas deferens, it was 1.17-fold increase. So there was significant increase in the MDA level. So reactive oxygen species might be playing a very important role in causing the azoospermia. So from these results, it can be concluded that ROS plays a very important role in the spermatogenic failure. Similar results could be seen in case of Serratia marcescens also, where we could find that significant increase in the MDA level in the right side of the organs, maybe testis, maybe epididymis, maybe vas deferens, and there was increase in the left side also, but it was lesser as compared to the right side. So in case of testis, it was fourfold, in the left testis it was twofold, whereas in case of epididymis it was 1.43 on the right side and 1.21 on the left side, and whereas in case of vas deferens it was 1.59-fold increase, and in case of left side it was 1.29-fold increase. So similar pattern could be seen in case of Serratia marcescens also. Almost similar results could be seen in case of Klebsiella pneumoniae also, where also we could find the significant increase in the MDA level on the right side of the organs, maybe in the testis, maybe in the epididymis, maybe in the vas deferens, although there was increase in the left side of the organs also, but it was lesser as compared to the right side. In case of sperm immobilizing microorganisms, Staphylococcus aureus, also there was increase in the MDA level, and this increase was again more on the right hand side as compared to the left hand side, and more increase was in the testes as compared to epididymis and vas deferens. So similar pattern was seen in case of the sperm immobilizing microorganism as we have seen in case of the sperm agglutinating microorganisms. The next experiment we performed was the catalase activity, and here H2O2 was used as the substrate, and we used the tissue homogenate to find out the level of catalase, and disappearance of H2O2 by catalase was measured at 240 nanometers spectrophotometrically. Absorbance change of 0.01 as unit per minute defines 1 unit cat activity. When the level of catalase activity was observed in the tissue homogenates, again it was found that there was significantly increased level of cat on the right hand side as compared to the left hand side. Again the increase was much more in the testes as compared to the epididymis and the vas deferens. So as we have seen in case of the MD level, catalase activity also followed the same pattern. When we check the catalase activity in the tissue homogenates of the mice administered with Serratia marcescens, again we got the same pattern, that the level was significantly high on the right side of the organs as compared to the left side of the organs. And moreover, the increase was significantly high in case of the testis as compared to epididymis and vas deferens. So it also followed the same pattern as we have seen in case of E. coli. When we check the catalase activity in the mice administered with Klebsiella pneumoniae, we could find that again the level of catalase activity was significantly high on the right side of the organs as compared to the left side of the organs. And again we found that the level of catalase in right testis and right epididymis was almost same, but it was significantly higher as compared to the vas deferens. In case of sperm immobilizing Staphylococcus aureus administered mice, when catalase activity was checked, again it was following the same pattern, that the level was significantly high on the right side of the organs as compared to the left side of the organs, and again the value was higher in the testes as compared to the epididymis and vas deferens. The next experiment we performed was the peroxidase activity to measure the ROS level, and peroxidase activity was determined by the amount of H2O2 utilized in the formation of tetraglycol. So POD level was estimated in the tissue homogenates, and the absorbance was measured at 470 nanometer, and change in absorbance of 0.01 as units per minute defines 1 unit POD activity. When the level of POD was checked in the tissue homogenates of the mice administered with sperm-agglutinating E. coli, again we could find the same pattern, that the level was high on the right hand side of the organs as compared to the left side of the organs, and we could find that the level was same in the testes and epididymis almost, whereas it was less in case of vas difference. Similar pattern could be seen in case of Serratia marcescens also, where the level was high in the right side of the organs as compared to the left side of the organs, and level was high in testes as compared to epididymis at vas difference. In case of Klebsiella pneumoniae administered mice also, when we checked the POD level, again the pattern was same. It was higher on the right side of the organs as compared to the left side of the organs, and more in case of testes as compared to epididymis and vas difference. So from all these results, we can say even the POD activity was also more in the right side of the organs as compared to the left side of the organs, and the level was more in the testes as compared to the epididymis and vas difference. In case of Staphylococcus aureus administered mice, again when we checked the level of the POD in the tissue homogenates, we could again find the same kind of the results, that the level was higher on the right side of the organs as compared to the left side of the organs, and again the level was higher in testes as compared to epididymis and vas difference. The next experiment we performed was the measurement of antioxidant level by the method of reduced glutathione. And here we took the 500 microliter of the supernatant from the tissue homogenates of the mice administered with different microorganisms, and this was then mixed with sulfur salicylic acid to precipitate out the proteins, and then it was centrifuged and the supernatant was taken. And glutathione in the supernatant was measured by subjecting it to oxidation by dithiobis 2-nitrobenzoic acid, and this resulted in the yellow colored complex, which was then measured at 412 nanometer, and GSH was expressed in terms of micromole GSH per gram of tissue using the molar extinction coefficient of 13.6 per millimole. From the results, it could be observed that in case of sperm-agglutinating E. coli administered mice, when we estimated the level of reduced glutathione, there was significant decrease in the level in the right organs as compared to the left organs, and again the maximum decrease was in the right testis as compared to right epididymis or right vas difference. So from the results, it can be seen that the antioxidant level goes very low as compared to the control group. So from these results, it can be seen the ROS level goes high and antioxidant level goes low, resulting in more severe condition. Maybe this is one of the mechanism by which the microorganism might be leading to azoospermia. Similar type of results could be seen in case of Serratia marcescens also, where also we could find the significant decrease in the level of reduced glutathione on the right side of the organs as compared to the left side of the organs, and again the maximum decrease was in the testes as compared to epididymis and vas difference. In case of sperm-agglutinating Klebsiella pneumoniae also, we could see the similar kind of the results. Again, there was significant decrease in the level of the reduced glutathione in the right side of the organs as compared to the left side of the organs, and again the decrease was maximum in the testes as compared to epididymis or vas difference. In case of sperm immobilizing Staphylococcus aureus administered mice also, similar results could be seen, where the level was significantly decreased in the right side of the organs as compared to the left side of the organs. Again, the decrease was maximum in the testes as compared to epididymis and vas difference. Antioxidant activity was also checked by measuring the level of superoxide dismutase in the supernatant of the tissue homogenates of the mice administered with different microorganisms. As we know that superoxide dismutase converts nitroblue tetrazolium into formazole crystals which are insoluble, and they can be solubilized by the solvent hydroxylamine HCl, and that results in the purple blue solution, and the absorbance of this purple blue solution can be measured at 560 nanometer. So measuring superoxide dismutase activity, we recorded the change in absorbance at 560 nanometer for 2 minutes at successive intervals of 60 seconds, and superoxide dismutase activity was measured according to the formula given as follows. From the results, it can be observed that there was again decrease in the superoxide dismutase activity in the right side of the organs as compared to the left side of the organs, and again there was significant decrease in the right testis as compared to epididymis, and vast difference. So antioxidant activity checked by superoxide dismutase activity also showed the same results as we have seen in case of the reduced glutathione. Similar pattern was seen in case of the Serratia marcescens also. Again, there was significant decrease in the right side of the organs as compared to the left side of the organs, and again the decrease was much higher in the testes as compared to epididymis, and vast difference. In case of Klebsiella pneumoniae also, there was significant decrease in the SOD level in the right side of the organs as compared to the left side of the organs, and again decrease was much higher in the testes as compared to epididymis, and vast difference. Similar kind of results were seen in case of the tissue homogeneates of mice administered with sperm immobilizing Staphylococcus aureus, where the level of SOD was much lower in case of the right set of organs as compared to the left set of organs, and more so in case of testes as compared to epididymis, and vast difference. All the present results, they can be summarized that the level of MDA, catalase and peroxidase activity goes significantly high in case of the test group as compared to control groups on both the sides of the organs, right side as well as left side, whereas the level of reduced glutathione and superoxide dismutase, it goes significantly low in the test group as compared to control group. So in conclusion, the increase in the level of MDA, catalase and peroxidase activity in the test group indicates a significant increase in oxidative stress, and conversely, the marked reduction in reduced glutathione and superoxide dismutase levels points to a compromised antioxidant defense system. So all these results, they can be hypothesized that oxidative stress might be playing a very important role in causing the azoospermia. Further, to find out what are the changes that are taking place in the testes, histopathological examination was carried out, and we only used right testes for the same experiment because in the previous results we had found that microorganisms, they had the most important impact on the right testes as compared to the left testes. So when histopathological examination of the right testes was carried out, we could find that in the control group no clinical sign of histological alterations as regular spermatogenesis was observed. Testis was normal, showing clusters of spermatozoa in the center surrounded by layers of germinal cells. Whereas in case of mice administered with E. coli, it was found that there was degeneration and necrosis in the germ cell layers. Testis demonstrated no mature spermatozoa and spermatids, which reveals azoospermia. And in case of the mice administered with Serratia marcescens, testis revealed very few mature spermatids that indicates severe hyperspermatogenesis. And in case of mice administered with Klebsiella pneumoniae, Leydig cells along with the basement membrane of seminiferous tubule was noticed, but was found to be thin as compared to control and revealed hyperspermatogenesis with few spermatids count. So histopathological examination also shows that the testes showed either the complete azoospermia or severe hyperspermatogenesis. The histopathological examination of the testes of the mice administered with sperm immobilizing Staphylococcus aureus showed reduction in spermatids in comparison with the representative control group. A reduced maturation of spermatids was detected, which indicated the condition of maturation arrest. Hence, a condition of mild hyperspermatogenesis was observed. So from these results also it could be concluded that sperm immobilizing microorganisms, they are less efficient in causing the azoospermia as compared to the sperm agglutinating microorganisms. In case of sperm immobilizing microorganisms, there was hyperspermatogenesis, but it was not as severe as in case of the sperm agglutinating microorganisms. The results of the present study can be summarized as follows. The administration of microorganisms was carried out by the intertesticular route exclusively into the right testes. In the test group, the bacterial load of microorganisms was recorded within the infected tissues of the male mice genital tract. Notably, a consistent dissemination pattern emerged in all test groups, extending from the right set of tissues, specifically from the right testis to right epididymis, followed by the right vas deferens, and on the left side from the left vas deferens to the left epididymis and to the left testis. In contrast, mice inoculated with PBS exhibited an absence of viable counts, underscoring the stark difference in bacterial presence between the two experimental conditions. Remarkably, the introduction of bacteria led to complete azoospermia, as evident by negligible sperm count on the right side for the test groups inoculated with SA microorganisms, and none of the seminal parameters could be evaluated. For Staphylococcus aureus sperm immobilizing inoculated test group, significant reduction in the sperm count on the right side was observed, exhibiting significantly reduced motility and viability percentages. Furthermore, only minimal presence of sperm count was observed on the left side for both SA and SI inoculated test groups. The few sperm detected on the left side were predominantly immotile or non-viable. Further, our study demonstrated that the intertesticular inoculation of sperm agglutinating and sperm immobilizing microorganisms in male mice leads to significant spermatogenic failure, thereby resulting in azoospermia. To further understand the mechanism of spermatogenic failure, the level of serum testosterone and reactive oxygen species was measured. Serum testosterone levels in male mice infected with microorganisms exhibited a prominent reduction as compared to control group levels. It is noteworthy that the observed testosterone values were notably below the established normal ranges for male mice. MDA level, catalase activity, peroxidase activity depicted significantly higher levels with respect to control groups across both the right side and the left side reproductive organs. Levels of reduced glutathione and superoxide dismutase decreased markedly in the test groups. The histopathological examination of the reproductive organ of all test groups showed altered histology in comparison to the control group, where normal tissue histology was observed. Results revealed that hypospermatogenesis was prominent in the testes of test group. These results show that reduced testosterone level and increased oxidative stress might be playing a very important role in spermatogenic failure, leading to azoospermia. Future prospects. Consequently, monitoring testosterone levels and assessing ROS balance may serve as valuable biomarkers in the diagnosis and understanding of azoospermia. These insights underscore the need for further research into the pathophysiological mechanisms underlying bacteria-induced reproductive dysfunction. As our understanding of medical science progresses, interventions focused on restoring hormonal equilibrium and mitigating oxidative stress represent promising avenues for enhancing male fertility and improving the well-being of affected individuals. Thank you.